Swayam Prabha. Digital India. Educated India.
In the last class we looked at how toxi pattern of different amino acids can be used for sequential assignments. So basically the idea is that each of the peaks of these residues such as the alpha peak, the beta peak and gamma peak, they have very regular pattern for each of the amino acids, 20 amino acids. So we call it a fingerprint. So each amino acid can be identified based on the chemical shifts in the aliphatic portion of the spectrum. So this is shown here. So you can see for all the 20 amino acids, you can see that there are very nice different patterns which can be utilized for matching the residues. So this is what we will use now as we go along in for sequential assignments. So you can see this is for glycine, there should be, as I said, there should be 2 peaks, sometimes you may get 1 or sometimes you may get 2, but they are not chemically equivalent at times. And then you can see here at alanine gives a very clear cut peak at 1.5 ppm and the alpha region at 4.5 or sometime even at 4. So as different amino acids have different characteristic patterns. So basically what we do next is that given the toxi spectrum, the 2D toxi spectrum, we have to now identify the different spin systems in the peptide. So remember a peptide is composed of a sequence of amino acids, and each amino acid as we saw can be denoted as a spin system. So that is what we will do. We will also look at the H alpha. So you can see one thing, if you notice, the H alpha regions are kind of not very distinguishable from each other. There is no particular pattern as to where the H alpha peaks belong compared to what you see this side. So here if you notice, here on this side, that is aliphatic beyond between 1 and 3 ppm, there are clear cut number of peaks which are different for each amino acid, okay, except here. So here you see, here these are called AMX spin systems. AMX spin systems are basically those which have only the beta carbon and 2 beta protons. For case of phenylalanine, tyrosine, histidine, tryptophan, you have what is called side chain aromatic spin system. But we will not look into all those when we go for sequence specific resonance assignments, in which we only look at the backbone and the side chain up to the beta protons or carbon or gamma carbons. Okay. So these are called long side chains amino acid. So that glutamic acid, glutamine, lysine, arginine, they belong to what are known as long side chain amino acids. So one thing, so coming back to this protons, what we see here is that there is no strict pattern, but this is the region which we will be using for sequence specific resonance assignment. So let us go back to this sequential assignments. So this is what we see here. So we saw the cozy pattern, we saw for threonine what is the chemical shift type of amino acids, what are the different patterns, and we looked at valine. So in the valine we saw that you have this amide proton, and then this is alpha, beta, gamma, delta, this is gamma 1, gamma 2, and this is a beta proton and this is a gamma proton, and this is 2 gamma protons. So this is the beta proton and this is the alpha proton. So this is something which we looked at, and as I said, this is a peak pattern which is characteristic for each amino acid. So now let us see how toxi can be used for spin system assignment. So this is what is shown here is a toxi cutout from just a schematic diagram. This is not from the peptide which we are going to look at. So what, as it says here, the toxi spectrum gives the entire spin system network showing peaks from the NH all the way up to the side chain protons along with single line along a single line. So you can see, if you look at this here, if you look at the single line here, that means if I look at a toxi portion, this is a portion of the toxi spectrum, this is not the full toxi spectrum. So for a portion of the toxi spectrum, you can see from NH, this is alpha, this beta, gamma, delta and so on for each amino acid, and this is what we will be using for sequential assignments. So, this is basically in the case of NOSI, what happens is we will see that later, but in a NOSI we not only see the spin system along this line, but we will also see the neighboring amino acid peaks also in this. So, right now let us not focus on this, we will go ahead and come back to this at the later point. So, this is basically what how do we assign the sequential peaks. So, you can see here, for example, you have a toxi peak which is showing for alanine, you have alpha and beta, this is the protons, and let us say you have a leucine. So, now I am looking at a dipeptide. Okay. So, when you look at a dipeptide region of a amino acid, what we are going to see. So, let us say that this is the NH of I, residue I, and this is NH of residue I minus 1. So, let me again illustrate you where we are actually focusing on here. So, if look at this is a toxi spectrum. Okay. So, this is diagonal. So, we are looking at toxi, and this is between 0 to 10 ppm, and this is between 0 to 10 ppm. Okay. So, the amide region comes in this portion, this is 7 to 10 ppm. So, this is the amide region. Okay. So, what we are looking at in all these cases is basically this region. So, we are looking at a particular strip. So, we are looking at a particular residue. So, if we go back to the previous slide. So, if you see in this previous residue here, this slide we see here, we see this strip here, we can see a line of peaks belonging to one amino acid, and this is what we known as a strip. The strip because I can cut out a strip like this, and that will correspond to one amino acid. So, in that particular region, this is the toxi region, but again remember we are looking only at the amide region. So, 7 to 10 ppm, and this is the region where all the peaks are coming in the amide portion. So, if you look from the amide regions, that is the NH, the amide means NH. So, for a given residue, let us say I, we have identified suppose the spin system, and we know it is a leucine spin system. Okay. So, right now in this class we are going hypothetically through a schematic drawings, we are not looking at a real peptide. We will see the real peptide in the next few slides, I mean the data from an actual peptide, but so right now it is all in a schematic mode. So, if you see if you look at an amide NH region of amino acid which is somewhere here in this region of the spectrum. So, this is the total toxi spectrum, and this is the amide region here, and in particular one, let us say one particular spin system we identify as leucine. Okay. And there is another spin system where we identify as alanine. Okay. So, these are two different spin systems, both of which are present in the toxi region. Okay. So, now we want to figure out which is coming after which. So, you can see as per the letters written down text here, NH is residue I and NH is this residue is I minus 1. That means alanine is coming before leucine. Okay. But how did we find that out? We can find it out from the NOSI. So, what will happen in a NOSI is that the two different spin systems which are there in toxi, that will be combined, you will see in a NOSI. Okay. So, if you look at the NOSI NH region, so if you look at the NH chemical shift of the leucine, the same chemical shift. So, you see this is the leucine spin system, okay, and there is this residue I, which is NH value is somewhere, let us say 7.1. So, let me again write down so that is clear. So, let us say that this one is 7.5. Let us say, sorry, I will erase this. So, let us say that this is 7.5 ppm, and let us say this is 8.1 ppm. Okay. So, this is the amide chemical shift. Now, here this particular region is going to be, we are looking at is 7.5. Okay. So, this is same as the leucine. Okay. So, now in this particular region here, when it is 7.5, that is the same chemical shift, but this is coming from a TOXI spectrum and this is coming from a NOESY spectrum. So, in the NOESY what you will notice is that you will notice the leucine chemical shift which are shown in the red color. Okay. So, the leucine chemical shifts are coming in the red color in the same ppm value as in the TOXI, and this is not surprising because this is nothing but the same amide chemical shift as the TOXI. So, TOXI and NOESY are giving the same correlations, they always give the same correlations, but additionally now if you see, so this is what is shown by this line. Now, what you will see, these green peaks here, they correspond to alanine chemical shifts. So, this is the beta proton of alanine and this is the alpha proton of alanine, and also we are getting a correlation to amide. So, what is happening here is that in the NOESY spectrum you not only see the correlations of the TOXI from the same residue where you are looking at. So, for example, I am looking at this line which is 7.5 ppm, and the same line 7.5 ppm in TOXI will give me a spin system, and therefore, that spin system is completely observed here, means it is present in this line also. But additionally in this line we will also get the correlations from I minus 1. Okay. So, this is where the most important aspect of NOESY and TOXI combination comes into play. The idea here is that in a TOXI you see individual spin systems, that is individual amino acids, but in a NOESY you get a combination, you get a dipeptide, you get two amino acids together. And why is this happening? This is happening because the neighboring amino acid is not very far away from the amino acid which we are looking at. So, we are looking at a residue I, and I minus 1 is not far away, it is within 5 angstrom. So, if you draw this picture again here, so we are looking at a dipeptide like this. We are looking at a dipeptide like this. This is some side chain, this is amide, this is another side chain. So, what we are seeing is we are seeing a correlation from here to this residue in a NOESY, and correlation from here to this residue in a NOESY. So, the NOESY gives both these correlations. Okay. This is what happens in a NOESY, because this is what I have written as 7.5 ppm in this picture. So, I am getting correlations, that is connectivity information from not only this amide, this portion here, to the self means to the residue I. So, I will call this I am also getting correlation to I minus 1 in a NOESY spectrum, but not so in the TOXI. So, we have seen this in the previous class, that in a TOXI you get correlation only from the self amino acid, that is only from its own amino acid, this is I, but you do not get I minus 1 correlation which is present only in the NOESY spectrum. Okay. So, now we can see what is the advantage is that I am able to get a correlation between I and I minus 1 between two amino acids. So, this actually can be used for what is called as sequential assignment. So, what is sequential assignment? Sequential assignment is the process in which we establish a link between two neighboring amino acid. So, if I go from I establish a link correlation to I minus 1. Now, if I go to I minus 1, that is this NH here 8.1 ppm, and if I go to the NOESY of that, so that is not shown here, but if I go to the NOESY of that residue, then I will get correlation to I minus 2. Similarly, if I go to I minus 2, I will get correlation to I minus 3, and so on. Similarly, if I go to I plus 1, which I do not know which is I plus 1, any residue, suppose there is a residue I plus 1, that should show a correlation to the residues of the spin system I. So, in the NOESY. Okay. So, this is how we form what is called a sequential link. So, we are essentially establishing a connectivity from I to I minus 1 to I minus 2 and so on. Similarly, we are going in this direction to I plus 1 will give me to I plus 1 will give me to I plus 2. So, we start from an arbitrary I, arbitrary which we do not know what residue it is. So, we start from an arbitrary I and then we move in both directions, and basically that will give me the sequential assignment. So, that is the goal of this whole exercise, is that I am trying to establish sequential resonance assignments, and that is done by which starting from an arbitrary spin system I, which we do not know what residue it is, we can identify it based on the toxi that it could be a particular amino acid type. Then starting from I then look for a correlation to I minus 1 in the nosy. Then I will also see in nosy which other spin system is giving correlation to I. That should be then I plus 1, because I plus 1 will give it to I. So, one thing you see in this case, in this experiment, in this discussion, is that we are only unidirectional, it is a one way path, meaning I am only looking at correlation from I to I minus 1. That means, in a nosy spin system, in a nosy peak, in a nosy spectrum, for a given residue I will get correlation only to I minus 1. And why is that so? That we will see that later. You can also get a feel from this picture here that this amide residue is close in within 5 angstrom for the nosy to be observed in to the sequential I minus 1. But if it has to see correlation to I plus 1, that is little far away, it goes beyond this here, because there is a amide bond here and then comes the side chain. So, typically the I minus 1 is the closest for the amide proton of a residue I, but I plus 1 is not so close. So, therefore, any spin system I in a nosy will show correlation to mainly I minus 1 spin system. Okay. So, if you want to see I, you have to go to I plus 1, then I plus 1 will give correlation to I, and I plus 2 will give to I plus 1. So, you see we are going in the direction from I to I minus 1. So, this is basically the point in the entire exercise when we use nosy for sequence specific resonance assignment. So, let us see now what we will do is we will only focus on this portion in the next slide, and from there we will see how we can do the sequential assignment from by going from I to I minus 1 to I minus 2 and so on, or I to I plus 1, I plus 2 and so on. So, that is basically our goal. Our goal is to establish a link between the spin system I and other spin system in the molecule, the peptide. So, this is what I was showing, that we are going to look at only the alpha region. Okay. The idea is that this alpha region will be used for establishing the connectivity. This means I and I plus 1 or I and I minus 1 at any point, and then once we identify the connectivity, then we will go to the side chain here to identify what amino acid it is. So, let us do this exercise. So, this is what is shown is again as a schematic. The real example, as I said, we will take it up subsequently. So, this is a 2D nosy spectrum, and if you again you should keep careful attention to the point that we are looking at a very small region of the whole spectrum. This is not the full spectrum. The full spectrum will go from 0 to 10 ppm on both the axes, but here we are only looking at amide region, which is 7 to 10 ppm, and we are looking only at the alpha region, H alpha, and if you recollect from the previous slide, it comes somewhere between 3.5 to 5 ppm. So, this region what you are seeing on the y axis is actually only from 3.5 to 5 ppm. So, now what we will do in a rosy, taking a nosy, or it can be a rosy, it does not matter, both experiment give, remember, the same correlations. So, I will take a rosy or a nosy spectrum, and then I will zoom into a particular spin system I, and remember again this is an arbitrary, I am just looking at some peak in the rosy spectrum and I will call that as I, and this is in the H alpha region. Now, from the toxi I will know the spin system which is corresponding to I, but in the nosy I will only look at the correlation it is giving to another peak. So, in a rosy spectrum it will give me a correlation to I minus 1. Then I minus 1, if I go more further, it should now correspond to some this is the H alpha of I minus 1. So, when I actually look at the self chemical shift of I minus 1, which is here, then at that place not only this I minus 1 will be there, this peak, but it will now show correlation to I minus 2, because I started from a residue I which is arbitrary and I found its I minus 1, because in a nosy I get both the correlations. Now, I searched along this line, horizontal line, and saw where this comes, and I found that it comes here, and that vertical line now corresponds to I minus 1, because it is the self, means its own, and it was a sequential. So, when the self of I minus 1 is found, there if I look, I may get another peak which will now belong to I minus 2. So, remember this peak can come either down or up, that is not important. What the important here is that this along the horizontal line you go, you find the peak corresponding to I minus 1, then you go along the vertical line and you get I minus 2. Now, similar if I continue this, I should go along this line, then I will get somewhere where these two peaks coincide, and that means that peak here, this line, vertical line, should belong to I minus 2, because I minus 2 will give correlation to I minus 2 self, that is called its own, and it will give a correlation to sequential, which is now in I minus 3. So, you can see here what we are doing essentially, you are starting from a self, means some residue I which is its self chemical shift, its own chemical shift, going along the vertical line, finding in this line within this region. So, remember we are only focusing on this region, and this region is called the H alpha region. So, within the H alpha region I go up, I find I minus 1 which is a sequential. Then I go along the horizontal, then I see where this I minus 1 comes in this spectrum, and I notice that it comes somewhere here in this vertical line, and that vertical line now becomes I minus 1 NH, means the amide of I minus 1. So, again remember we are looking at the amide portion. Once I find that I minus 1 NH, again I search either up or down to see if I see another peak, and if I get another peak, that peak should now belong to I minus 2, because in a nosy I gives only to I minus 1, I minus 1 gives you only to I minus 2, and so on. So, now once I got I minus 2, I will go horizontal along this line, find another spin system where this is present, and that spin system now should belong to I minus 2, and its sequential will come as I minus 3. So, this is the manner in which we carry out sequential assignment. We are essentially linking, again remember the word link, we are linking or forming a sequential correlation between a residue I and its sequential I minus 1. So, now let us come to this place here. So, suppose when I started arbitrary from a residue I or a peak I, from the toxi, as I said, we will know the spin system, and suppose that spin system belongs to a residue alanine. It looks, suppose it is an alanine spin system, and let us say I minus 1 which is along this line, vertical line, is, let us say suppose it is another spin system leucine, and I minus 2 let us say is glycine. So, what we have done now based on this toxi, we have identified the spin system corresponding to I minus 1, I minus 2, and we have also formed a link between them using the nosy spectrum. So, what we have to do now is search in our sequence of amino acid if there is such a system, if there is such a string of amino acids. This is our peptide sequence which is shown here. So, suppose this is our peptide sequence. Now, I will search for a residue which is I should be ala, L should be I minus 1 glycine, that means it should be a GLA spin system. So, there is a typo here. So, let me correct this. So, this is basically this will be, so I will call other way around. So, let me say that this I is glycine, I minus 1 is leucine, and I minus 2 is alanine. See, it does not matter because this is just a schematic right now. So, we are only looking for a tripeptide because we have formed a tripeptide connection here. So, that means I am looking at a system I is glycine. So, alanine, leucine, glycine. So, this is I minus 2, this is I minus 1, this is I. So, what I have done here is based on the TOC, I have identified a tripeptide TOC and NOZ, identified a tripeptide which is ALG, and now I am trying to look for this tripeptide in my peptide sequence. Okay. So, the peptide sequence you can see, I can see that there is a ALG here, and which is what I got from my linking of the residues. So, this is how we say that we have assigned this spin system. So, we can say now that we have assigned this tripeptides ALG. Okay. So, remember one thing is that yes, ALG, each of them is unique amino acid. So, you may have to still establish a link between them, only then you can say that I have completed the assignments. Without establishing a link between amino acids like we do based on NOZ and TOC, we cannot say that we have done the sequence specific resonance assignment. So, sequence specific resonance assignment, now if you recollect, it is based, there are two things we have to achieve in that. Number one, we have to do spin system identification, means from the TOC we have to identify all the amino acids, what amino acids they belong to, and from the NOZ we have to establish a sequential link. So, let us take an example, a real example of a peptide and with data, and then go through this exercise again with real experimental spectrum. So, this is the peptide which we are going to look at how we can assign using the approach which we have learnt just now. So, this is a 1D spectrum of this MFYGL peptide. So, M is methionine, phenylalanine, tyrosine, glycine and leucine. So, now looking at this 1D spectrum, it is not possible to assign because there are a lot of peaks and which are overlapped and the multiplex structure is not clearly visible. So, we will basically have to use the 2D approaches that we have learnt in this class. So, let us look at the different regions of this 1D spectrum. So, this is called the methyl region because this all the peaks belonging to the methyl groups will come between 0 to 1 ppm, 1.5 to 2 ppm. So, what are the methyl amino acids here? It is basically leucine and there is a methyl group in methionine. So, those are the peaks which will appear here. Then the next region is called as the H alpha, H beta, H gamma. This is the methylene region. In the methylene region you get CH2 peaks and that comes typically between 2.2 to 3.5. Then comes the H alpha, that is the backbone H alpha region of all the amino acids, and there are invariably always between 3.5 to 5 ppm depending on the secondary structure. So, this is going to be a very important region which we will focus for sequential assignments which we saw, and this is the aromatic region which comes between 7 to 7.5, and this is similar to what you see in the organic compounds. We saw that we also get aromatic peaks in this region, and then comes the amide region. So, amide region is basically the backbone CONH amide and as well as side chain amide. Side chain amides are present in some amino acid such as asparagine, glutamine, they are amides, and side chain amines are also there in amino acids such as lysine. So, we are now going to focus on how we can use these two regions. So, essentially the H amide and H alpha region will be useful for us. So, let us go through the different 2D spectrum for this peptide. So, the first experiment you would record is the 2D proton-proton cozy, and as we have seen, this experiment basically correlates two protons which are close to each other. So, in this peptide we will call this side as the N terminus and this side as the C terminus. This is typically the nomenclature or notation which is used in peptide assignments or protein assignments. Now, one thing one should keep in mind is that when we have a peptide dissolved in water, in this particular example we have taken it in DMSO, but if you have water sample dissolved in water or D2O, what happens is the N terminal, the NH3 plus. So, this residue will not have a peptide bond, it is peptide bonded to this through the carboxylate group, but the N terminal NH of this is free and that is NH2 or NH3 plus, and that exchanges very rapidly with the solvent, and because of that we will not get any peak for NH peak for this amino acid. This is exchange part, we will see that in the next part of the course when you go to the advanced topics, but this is typically one has to keep in mind. But when it comes to DMSO solvent, DMSO is aprotic and there we do not need to worry about this problem. So, now we will in the next class we will basically see how these correlations, that is NH alpha correlations, can be used for sequential assignments as we have seen in case of a schematic drawing.